let us talk about digestion, the duodenum. Duodenum is a part of a small intestine. Don't forget that. Duodenum is part of a small intestine. So when I talk about duodenum, I'm looking at the top of a small what? Top of a small intestine. So we are beginning digestion in the small what? Intestines. Now, once the food has reached the duodenum, now the presence of food in the duodenum stimulates the duodenal walls to release an hormone known as secretin. Now, secretin also stimulates the liver to produce what we call the bile. Bile is secreted to the duodenum via what we are calling the bile duct. Then the bile, which has been secreted, this bile emulsifies the fats. This one breaking the fats into small what? Droplets. Then the same secretin also stimulates the pancreas to release what we are calling the pancreatic what? Juice. This juice also passes through what we call the pancreatic what? Duct. Now we are going to see a mixture here. We have bile, we have pancreatic juice. Now let us try to see the components which are there in those secretions. Pancreatic juice, we are seeing that the pancreatic juice members contains what we are calling pancreatic amylase. The pancreatic amylase converts the starch to maltose. It is acting the same way like the salivary amylase. We also have what we call the trypsin. Trypsin, this one converts the proteins in the food to peptides. This one is also acting in the same way like the pepsin. Then we have even the lipase. This lipase converts the fats to fatty acids and the glycerol. Now, you can see that the stomach adds its enzymes. The stomach adds its enzymes. And we are seeing that even duodenum is having its own what? Enzymes. Now, these enzymes acting differently. They are specific in nature. And they also work in different pH medium. Now, the pH medium in the duodenum is going to be created by the bile. So let us try to look at the roles of bile in the duodenum. Bile, one of the roles of the bile stops the work of pepsin because it is alkaline. It neutralizes the hydrochloric acid from the stomach and makes the condition alkaline. The same bile activates enzymes in the pancreatic juice by providing an alkaline medium. That one can be illustrated properly by the diagram showing the relation between the stomach, the liver, and the pancreas. So therefore, in the duodenum, we are going to mainly reflect on the pancreatic amylase, the trypsin, and lipase, which are in what we are calling the pancreatic juice. The bile is just containing some alkaline salts, which are providing alkaline what? Alkaline medium. So it is only neutralizing the acidic food from the stomach into a medium which is what? A medium which is alkaline. We can continue with our small intestine and then we talk about digestion, the ileum. Members, in the ileum is just a part of the gut where final digestion of food takes place. Every food substance which has not broken down, its digestion will end at this point. So presence of food in the ileum stimulates the walls of the ileum to secrete what we are calling intestinal juice. This intestinal juice, some biologists can even complicate it by calling the succus entericus juice. But what matters is to know that it is intestinal juice. The intestinal juice, or the succus entericus juice, contains the following enzymes, which are very, very vital in now finalizing the digestion of the food into simpler materials that we need in our blood. Remember, our journey is to make the food. We have taken a potato, a big potato. It is to get glucose from the potato. You have taken a millet. Then you have taken a portion. Those ones, all of them are going to give us glucose. You have taken a fish, you have taken meat, you have taken beans, cowpeas. The final product is going to be what we are calling amino acids. You have taken a cooking oil, you have taken fats for example, in mutton, beef. Final digestion is amino acids as well as fatty acids and glycerol. Now we are seeing that presence of the food is causing now release of intestinal juice. The intestinal juice contains the following. We have the first enzyme there known as peptidase or lepsin. This one converts the peptides to amino acids, completing digestion of the proteins. Then we have what we call the lipase. The lipase converts the fats to fatty acids and glycerol, completing digestion of the fats or the lipids. We have the sucrase. The sucrase converts sucrose or cane sugar to glucose and fructose. Then we have what we call the maltase. The maltase converts the maltose or the malt sugar to glucose. Then we have what we call the lactase. The lactase converts the lactose or the milk sugar to glucose and galactose. So you are seeing that we have only the final enzymes. These enzymes are just breaking down the food, the smallest unit. Remember, food, we said, being broken here is having a complex structure, what we are calling a polymer. And now we are seeing we are forming monomers. That is now chemistry language. So we are turning the polymers like a starch to monomers like a glucose. Polymers are very big. They cannot pass via the small pores of the villus. So we are seeing our small pore only accommodating small structures, which are calling like glucose. The glucose is the final end product of carbohydrates. Then the amino acids, those are the final end products of the proteins. Then we can also look at fatty acids and glycerol. Those are the final end products of what we are calling the lipids. Now, after understanding that process of digestion, so in summary, digestion of food substances in the human alimentary canal will be as follows. Digestion of proteins. First, protein food is mechanically broken down in the mouth by the action of the teeth during chewing or mastication. Then this is also termed as physical digestion. The food is then mixed with saliva, which lubricates the food. Then what happens to the protein food in the stomach canal? Remember, from there you are going to swallow. You are going now to food. If the food is mixed with saliva, we said the tongue allows the food into balls, and these balls will move by peristalsis in the what? To the stomach. Now we are seeing our food reaching the stomach members. When the food reaches the stomach members, now we are going to see that the presence of food in the stomach stimulates the release of the gastrin hormone. When it stimulates the release of the gastrin hormone, what we are going to see, we say the gastric hormone stimulates the release of the gastric what? Juices. Which gastric juices contain pepsin, contain renin, contain hydrochloric what? Acid. At the same time, we are seeing that we have the mechanical digestion in the stomach by churning the food. So the mechanical digestion of protein food is done by muscular walls of the stomach and by the rhythmic contraction of the muscles. The food contained in the stomach canal is churned up and thoroughly mixed with gastric juice produced by the stomach wall to the creamy liquid called chyme. And this is also physical digestion. Then the gastric juice produced contains the pepsin, which pepsin breaks down proteins to peptides, the renin, which coagulates the milk protein, casein, and then it can be attacked by pepsin, then the hydrochloric acid, which now provides the pH, which also kills the bacteria. Don't forget that. Then the food flows to the duodenum. In the duodenum, we are seeing that the pancreas secretes pancreatic what? Juice. Remember that one, it is stimulated by secretin. Don't forget that. Then the pancreas is going to produce what? It's going to produce pancreatic what? Juices. The pancreatic juice contains what? Trypsin. Trypsin does what? Breaks down the proteins to peptides. Then the salts. At the same time, we have the salts in the bile, which is produced by the liver. Remember, at the same time, secretin stimulates the liver to release bile, which passes via the bile ducts and mixes with the pancreatic juices. Then what are we seeing here? The salts in the bile neutralizes the acid food or the chyme from the stomach to provide the alkaline media. Then we are seeing there enzymes doing the work. You have enzymes like pancreatic amylase breaking down the remaining starch to maltose. Then you have which other enzyme? Trypsin is also there doing the work of breaking the remaining proteins to what we call peptides. Then you have what we call the lipase. Busy doing what? Breaking down the fats into fatty acids and what? Fatty acids and glycerol. Simple as that. Then from there, the food will continue to the ileum. Now in the ileum, the presence of the food now will stimulate the ileum wall to secrete succus, entericus juice or intestinal juice. This juice also contains the final enzymes which we talked about that are going to act on the proteins. And mainly we have what we call peptides. This one is going to break down the peptides to what we call the amino acids. Then amino acids will go where? Will pass via the villus into our bloodstream. And then when it reaches our bloodstream, we are going to see assimilation being put into use, like forming our structures, what and intestine. The large intestine is simply having no enzyme at all, but having a lot of bacteria that digest now part of the liphages or the fibers. Liphages are very hard to be broken by the other enzymes, but the bacteria can do it, like the sucrase bacteria can break the cellulose in the fiber. And the biggest role of the colon, that one is to absorb water from the food which is undigested. So this explains why you find that the food is very, very hard as it is passing out via the anus. So we are seeing that the mucus is secreted by the mucus secreting wall and is used to lubricate the pathway for the solid food to pass as feces. So mainly our colon is to absorb the water, and then the food which is solid is allowed to move downwards by peristalsis until when it is expelled via the anus, a process known as digestion. That is when the body has already picked all its digested food materials